Hey everybody, welcome back to not back to chapter 20, welcome to chapter 20, back to microbiology. Let me just get this started up. All right. Okay, so chapter 20, you know the deal with this one. What we're going to be doing is the same as what we did for 18 and 19, but for the cardiovascular system. So you guys know the circulatory system pumps the blood all around the body. The heart is the one that's actually being the muscle that actually moves the blood. The blood vessels carry it. And you know, you've got veins and you've got arteries and then the capillaries. So on the next slide, I forgot about that. And basically that's how oxygen is going to get to everything, and it's going to carry away the carbon dioxide and waste products. So the arteries, remember, arteries go away from the heart, and the arterioles are the small ones. Veins carry blood towards the heart, and the venules are the smaller ones. And then we have the capillaries that are going to connect the venules and the arterioles. Basically, that's where a lot of that exchange is going to be going on. Then we know the lymphatic system, but it is part of the cardiovascular system. Remember, as far as, you know, moving things back towards the heart, that's going to happen with movement of the body through the actual lymph system itself. But then moving out away from the heart, essentially, it's going to all be part of the whole cardiovascular system. And not sorry, I have a little thing. I'll try not to play with that while I'm giving this talk. It's making noises. Anyways, you guys remember how that moves, and that we have lymph vessels, we have lymph nodes, we have the spleen, and that whole thing essentially works as a means to serve the immune system, collect fluid that has left the blood vessels and enter the tissues. We're going to bring it back into the circulatory system, filter the whole system, and return it all back to blood. Sorry, my nose itches. Okay, so we will need to protect the cardiovascular system from infection. Essentially, you don't want what provides you with oxygen to get, infected and get ruined. So the cardiovascular system is highly protected from microbial infection. Microbes that invade the system gain access to every part of the body and can affect every system, causing systemic infections all over the body. And what defenses does the blood have in place? Well, obviously that's where all of our white blood cells are going to be. So that should be what's providing you the most protection, as well as access into those lymph nodes and spleen and everything like that. There's going to be a lot of white blood cells like the lymphocytes, the B and T cells, as well as phagocytes, right? So medical conditions that involve the blood have emia at the end. We have viremia, viruses. That includes viruses that cause meningitis, fungemia, fungi in the blood, bacteremia. If we have any bacteria in the blood, that's bacteremia. But if the bacteria is flourishing and growing and, going to be causing disease as a result of that, that's septicemia, and that can lead to decreased blood pressure and septic shock. Like the nervous system, it should, we consider the cardiovascular system and the lymph system to be closed, essentially. So there should not be any microbes colonizing that. Microbes may be present transiently, but they shouldn't actually be, taking hold there. So that's fine. We're good with that. You guys know how these charts work. You're more than welcome to use these as study aids, but we're not going to go over this one in any detail. So I'm getting into the diseases. Start with endocarditis. That's inflammation of the inner lining of the heart. It can be acute or subacute, essentially. You might not know that it's what's going on. Damage to the heart valves can occur, or to the prosthetic heart valves if you've had a new heart valve put into your heart. And then when that happens, essentially damage to those valves from cholesterol or, a lot of times it's cholesterol, right? So plaque buildup and everything like that, or any inflammation due to maybe prior infections or something like that. But anyways, you have damage to your valve, and that can be like the starting point of how endocarditis can come out to be. It can also be caused by vascular trauma or circulating immune complexes. Essentially, we start out with the inflammation in the inner lining of the heart, and then that's going to be, sort of want to say home for infection. We can breed infection, make it the area a little bit more friendly to infection when it shouldn't be. All right, so if you have endocarditis, acute endocarditis, symptoms would include fever, anemia, abnormal heartbeat, symptoms of a heart attack in general could be felt. You might have abdominal or side pain, Janeway lesions, which we can see down below on the left, bottom left, Osler's nodes, and splinter hemorrhages, which is on the bottom right. And we have symptoms of acute endocarditis. These are similar to, I just realized something. Sorry, symptoms of subacute endocarditis. Similar to what we see with endocarditis, but develops slowly, it's less pronounced. We also might see enlarged spleen, clubbed fingers, and clubbed toes as well. All right, septicemia occurs whenever organisms are multiplying in the blood. We might see fever, altered mental state, shaking, chills, and gastrointestinal symptoms as a result. We also might have the presence of increased breathing rate, respiratory alkalosis, low blood pressure resulting from loss of fluid from the vasculature. This can be caused or be, I don't say be caused, this can be linked with endotoxic shock if you have an infection caused by gram-negative bacteria and they're releasing endotoxin. This will stimulate a massive inflammatory response that'll be mediated by cytokines. Just think of cytokines as being like messengers and telling everybody to, come to arms. This will lead to a drastic loss of blood pressure, again, leading to shock. And gram-positive bacteria have a similar series of events when fragments of their cell wall are released into the bloodstream, because remember, endotoxin is part of a lipid essentially for gram-negative bacteria, but gram-positive bacteria, they still got that whole big, thick gram-positive associated thick cell wall of peptidoglycan, and our body sees peptidoglycan as, itself could potentially be bad, especially when it's fragments of it. Shouldn't be fragments hanging out there, right? So it has a similar reaction to what we see with endotoxic shock. Moving on to the next thing is the plague. And yes, we are talking about the Black Plague, something we're very familiar with. These are all like the significant outbreaks back in the day. Essentially, we're familiar with this. We all know about this, plague doctors and all that. What causes the plague is a bacteria called Yersinia pestis. It is a gram-negative rod. Its reservoir is in rats, ground squirrels, and prairie dogs, sort of rodent type mammals. And the vector is Xenopsilla chiopsis, that is fleas. So fleas will bite the rats. The rats had Yersinia pestis. So the flea is now carrying blood from rats that have Yersinia pestis in them, and then they can go bite a human and then pass that on to people. Bubonic plague, we have bacterial growth in the blood and the lymph itself. And then we'll have development of buboes. There'll be a obvious visible swelling of lymph nodes, obvious and visible from the outside, like just from on the skin. Septicemic plague, which is going to lead to septic shock. That's going to be 30 to 50% of cases, and it has a 100% death rate. And then pneumonic plague, where it be transferred to the lungs. So this is just showing the route of transmission, where we have, the rodents, the flea bites it, and that can exist within as a reservoir in the normal nature. But eventually those fleas might bite humans and pass bacteria on that way. That can lead to bubonic plague due to transfer of the flea bite, the infected blood from within the flea into the person's bloodstream. And we would see presence of buboes and development of buboes and stuff like that. And then due to his infection, he could be passing that on from person to person, which it is. It can be transmitted from person to person due to development within the lungs, and that can lead to pneumonic plague. So this is buboes and the bubonic plague. And this is the Black Death. That's why we call it the Black Death, because of the bacteria in the bloodstream leading to damage to the blood vessels, especially in the capillaries, as well as blockages in the capillary area. We don't get enough oxygenation to those areas that have, the smaller vessels associated with them. And that usually includes the extremities. So they die and necrose. Okay, this is just how it's distributed in the world right now. We do have populations of prairie dogs that do carry the plague. It's just normal. It's always kind of been there, so no worries there. Just don't go, getting flea bites from prairie dogs, the fleas that were on prairie dogs. Next is tularemia. This is a zoonotic disease in the northern hemisphere, the normal hemisphere. It's transmitted by ticks or deer flies. That can also be transmitted by water and contact with animals that are infected, and inhalation of anything that could have the infected material in it. Symptoms will include headache, backache, fever, chills, malaise, and weakness. Signs will include ulcerative skin lesions, swollen lymph glands, conjunctival inflammation, sore throat, intestinal disruption, pulmonary involvement as well. I guess I would have put sore throat in symptoms. I'm just saying. So mortality rate is approximately 30%. I'm sorry, I really am. It's this that you're hearing the noise. I'm really trying not to play with it. Mortality rate is about 30% without proper treatment. I'm sure you guys can tell that I am one of those people that is a little bit ADHD. So tularemia is caused by a bacteria called Francisella tularensis. This is a gram-negative facultative intracellular parasite. And facultative intracellular parasite is something that can live within cells, but it doesn't have to live within cells. The infective dose is 10 to 50 organisms. What does that mean? That means it takes 10 to 50 of these bacteria cells to cause an active infection in an individual. This is listed as a pathogen of concern and for bioterrorism, for category A bioterrorism list. And rabbits and rodents are the reservoirs for it, as well as other mammals. Moving on to Lyme disease, this was first discovered in Old Lyme, Connecticut in the 70s. It is slow acting and it is progressive. About 70% of cases develop a characteristic bullseye rash, which is on the next slide. We have fever, headache, stiff neck, and dizziness associated with it, and it will eventually progress to cardiac and neurological symptoms. This can develop to crippling polyarthritis after several weeks to months of infection. This one's a difficult one, right? It's hard to diagnose, and it's one of those things that can be diagnosed a lot whenever it's not actually there. Just saying. But here are some incidents of the symptoms that are associated with it. And on the bottom left, you can see the typical bullseye rash that is associated with Lyme disease. So most common symptoms are going to be that rash, as well as arthritis, and other symptoms are a little less common. It's caused by the bacterium Borrelia burgdorferi. I like that name. It's a long spirochete, which means it's a spiral shape. It evades the immune system by changing its antigens. Remember, antigens are anything the immune system would be able to recognize or bind with. So it can change its surface antigens to things so that the immune system can't recognize it anymore and would have to keep reacting to it. So that it's possible that the immune response to this organism is part of the pathology with this disease. But again, this is a pretty poorly defined disease, and it's pretty hard to pin down. So this is just the life cycle of that organism. Essentially, you have it maintained within a rodent population. They get bit by ticks, the person gets bit by the tick by accident and is like an accidental host that way and develops Lyme disease. This is the distribution of Lyme disease across the United States. You can see that it is the largest number of cases are seen in the Northeast. This question asking, what is the causative agent of Lyme disease? Lyme disease is caused by Borrelia burgdorferi. That's the one that I like the name of. You remember Staphylococcus aureus causes a wide variety of skin diseases. Streptococcus pyogenes, typically associated with strep throat and scarlet fever, but it can cause also necrotizing fasciitis. And Francisella tularensis causes tularemia. All right, moving on to mono. It's caused by Epstein-Barr virus, which is a herpes virus. It has a circular DNA genome. It can become latent and hide out in the host cells, and it'll splice into the host cell DNA, and that allows it to evade the host's immune response for a long period of time. About 90% of the world's population has been infected with EBV. Symptoms are sore throat, high fever, and cervical lymphadenopathy. So very swollen, tender lymph nodes in the neck area. A long incubation period associated with it. You might have it for like 30 to 50 days before you start having that prodromal period pop up. And then fatigue and glandular fever are also associated with this pretty typically. It can eventually lead to the development of Burkitt's lymphoma. Burkitt's lymphoma is associated with Epstein-Barr infection, which is what causes mono. So this is also known as human herpes virus 4. Burkitt's lymphoma is a nasopharyngeal carcinoma. So up anywhere from the neck up into like the sinus area. I'm so sorry, my nose is so itchy right now. It causes cancer in immunosuppressed individuals. Typically, it's just immunosuppressed individuals. And we see this a lot associated with malaria infections as well as HIV. Typically AIDS, when it's progressed to AIDS. Anthrax is caused by Bacillus anthracis. Bacillus, what do we associate that with? Endospores. Do you remember what else causes endospores? Clostridium, right? So it's a zoonotic disease of herbivores. So normally exists in herbivores like cows. It causes cutaneous, pulmonary, and gastrointestinal forms of the disease. We've already talked a little bit about this when we were talking about like the skin stuff and everything. It's a gram-positive endospore-forming rod. It's aerobic. It's catalase positive. It forms a tripartite toxin, and the toxin is involved in the development of symptoms, edema factor, protective antigen, and lethal factor. Moving on to the hemorrhagic fevers. These are agents that infect the blood and the lymphatics and can lead to extreme fevers along with internal hemorrhaging. So a lot of bleeding associated with, especially internally. Sometimes you'll see that happening externally towards the end of the disease. There are 3 families of viruses usually that we will classify that have within those families hemorrhagic fever diseases. That's the arenaviruses, the filoviruses, and the flaviviruses. These are RNA enveloped viruses, so they have a lipid bilayer coating and an RNA genome, and they are classified, all of them, as BSL-4 pathogens. That means they are highly dangerous for handling. These include yellow fever, dengue, chikungunya, and Lassa fever, as well as Ebola and Marburg. These are just examples. So the yellow fever transmitted by mosquitoes, reservoir in monkeys. We have disruption of clotting leading to hemorrhage, eventually jaundice, and there is a vaccine available. Moving down to dengue fever, we see transmission via mosquitoes and has a mild infection, but we can also have the development of breakbone fever. Chikungunya, transmitted by mosquitoes, that which breaks bones is what apparently the word they're saying the word means. I remember I had brought this up previously and I said that I don't think that that's the same necessarily direct translation as the other one, but that's okay. We're not worrying about that. Anyways, chikungunya can lead to, and I played with the thing, sorry, an arthritic stance, and the disease can lead to a chronic infection. Lassa fever, 20% of cases are asymptomatic, but it can develop severe hemorrhage, and it is transmitted through rodent droppings. So here we have a focus on Ebola. It is a filovirus. Ebola and Marburg, they're very closely related. So the filoviruses are in the family Filoviridae. Symptoms are similar to yellow fever and dengue. We have capillary fragility, disruption of clotting, patients bleeding from their orifices and mucous membranes, massive internal and external hemorrhage, both. Mortality from Marburg has been cited around 25%, whereas mortality for Ebola has been cited around 97% mortality. There is no effective treatment for either disease necessarily, though there has been some indication that there may be success from passive immunity, which is giving the infected individuals antibodies from those who did happen to survive from the disease. There is a vaccine that is in use for this. How is it transmitted? It's pretty direct, right? So we do have some droplet, but it's not necessarily airborne, not like hot, like you cough. If you're right by me, you could potentially get it. I mean, that's pretty uncommon, but it's not like it's transmitted like through the air like tuberculosis is, okay? And it can also be transmitted through the blood. You have to pretty much have direct contact, though, to get it. Have you touched vomit, blood, sweat, saliva, urine, or feces of somebody who might have Ebola? No, you don't have it. You're not getting it. All right. Don't touch anybody that is sick. You can only get it from touching the blood or bodily fluids of someone who is sick or had died from Ebola, or being within very close proximity of somebody like that if they were like really like spitting it up and whatever. So don't go eating monkey bush meat, which is where this will often be started from. And don't go touching on fruit bats or any primates of any kind for a lot of reasons. I mean, they can carry other diseases besides Ebola, right? No sharing needles, and yeah, stay away from people who have Ebola. Symptoms include early on, we have fever and muscle aches, something you might not even be concerned about. The incubation period is 2 to 21 days where you wouldn't have any symptoms. Later on in the disease, we might have development of that severe hemorrhaging and organ dysfunction, but you otherwise might think you just have something like a flu at first. So when do you become contagious? Essentially, we have, where is it at? Approximately 8 to 12 days after exposure, whenever you start having symptoms, you are contagious. And even if you were able to, okay, so Ebola patients are most contagious at and near death. Pretty obvious, right? They've been growing that virus, they've been incubated, it's been happy, it's been growing. And all of that. So they're happy, the virus is happy. And so it's got a lot of it in your blood. So if you're bleeding out, man, you're going to be really super contagious. So that's the idea there of what it's talking about. So survivors improve their symptoms about 6 days after their symptoms start, typically, if they're going to clear it. So anyways, here's some of the past outbreaks of Ebola. Moving on. So what is the incubation period for Ebola? We had mentioned that the incubation period, which is the period of time that the virus requires in order to grow before you start seeing symptoms, is 2 to 21 days. All right, and the non-hemorrhagic fevers. These are diseases that are characterized by high fever, but they don't have any capillary fragility, and they won't have hemorrhagic symptoms associated with them. These are all caused by bacteria and include brucellosis, Q fever, cat scratch fever, or cat scratch disease, trench fever, ehrlichiosis and anaplasmosis, and then Rocky Mountain spotted fever. So brucellosis is also known as Malta fever, undulant fever, and Bang's disease. It's caused by a Brucella bacteria. That's a gram-negative coccobacilli. These live in phagocytic cells within the cells themselves, and the cells will carry the bacteria to the bloodstream and then lead to focal lesions on the liver, spleen, bone marrow, and kidney as a result of that. So it is considered a bioterrorism agent of highest concern. This is showing body temperature upon infection, so how many days after infection and what your temperature could be. So we have a dip in that body temperature down to the normal body temperature, and then we're going back up in the fever again, even after time. So Q is an interesting profile. Q fever stands for query because they couldn't figure out what was causing the disease. This is caused by Coxiella burnetii. It is a small pleomorphic gram-negative bacterium. It is an intracellular parasite, so it lives inside of cells themselves, the host cells, and they form an endospore-like structure, but not true endospores. These are transmitted by contact with the infected animals. Cat scratch fever is caused by Bartonella henselae. This is a small gram-negative rod. It is fastidious and will grow on blood agar. Infection is connected with being clawed or bitten by a cat, and it is present in about 40% of cats. Symptoms start at 1 to 2 weeks after a claw or a bite. And the cluster of small papules will start at the inoculation site, where they will have lymph node swelling. They'll become pus-filled, and about a third of patients will experience a high fever associated with infection. Trench fever is caused by Bartonella quintana. This is carried by lice. When lice feces enter a bite wound, that's how it would get transmitted, often by scratching at your scalp. Maybe not your scalp, but maybe homeless people's scalps. So this is endemic to Europe, Africa, and Asia, but there have been cases of urban trench fever. About 33% of homeless people in San Francisco carry it, and symptoms include a 5 to 6 day fever, leg pains, headache, chills, muscle aches, and a macular rash. And then endocarditis can develop as well. Next is ehrlichiosis and anaplasmosis. Human monocytic ehrlichiosis is caused by Ehrlichia schiefensis, and human granulocytic anaplasmosis is caused by Anaplasma phagocytophilium. These are tick-borne diseases. They are spread by exotid ticks, which are hard ticks. Signs and symptoms include acute febrile state, headache, muscle pain, and rigors, and patients recover rapidly, typically with no lasting effects. So it's not as bad as some of these other ones that we've seen. Babesiosis is a protozoan. So they said all of them were bacteria, but they are not. This one's a protozoan. Right, similar symptoms to Ehrlichia and Anaplasma, okay? And these are carried by ticks as well. Then we have Rocky Mountain spotted fever in the southeast and eastern seaboard of America. We have rickettsia, of America, and then we also have Canada, Central, and South America involved as well. It's caused by the bacterium Rickettsia rickettsii. It's transmitted by hard ticks as well, the exotid ticks. And signs and symptoms associated with this include chills, fever, headache, and muscular pain. Then we will have development of a distinctive spotted rash 2 to 4 days after the prodromal period, which is the initial showing of symptoms. If it is left untreated, the lesions will merge, they can become necrotic, and there can be cardiovascular disruption, thrombosis, hemorrhage, and it can lead to about 20% mortality if we don't treat it. If you do, you're still facing about 5 to 10% mortality if treated, and we do have this in Oklahoma. So this is just the areas where you could find these diseases. Moving on to Chagas disease, it is caused by Trypanosoma cruzi. It is a flagellated protozoan. It is the agent of sleeping, similar to the agent of sleeping sickness. The signs and symptoms associated with Chagas disease include, in the acute phase, we have mild to severe fever, nausea, fatigue, and then a chagoma at the bite site, which is like a swollen and inflamed area where the bite was. And then we have swelling of the eyelids if the bite was near the eyes. Okay, typically this organism that is associated with biting, the kissing bug, it will bite either near the eyes or near the nose or the mouth, typically of children while they are sleeping. And then when they defecate in the area, that's how this protozoan organism will be spread. The chronic disease can lead to inflammation and disruption of the heart itself. We'll have inflammation of the brain and association of inflammation with the intestinal tract as well. This is the kissing bug on the bottom right. And then on the top left, that's the actual organism that causes the Chagas disease, the Trypanosoma cruzi. Moving to malaria, signs and symptoms occur in 48 to 72 hour intervals. You'll have cycling of the symptoms, and that's a pretty common thing that we see associated with malaria. The symptoms themselves include malaise, fatigue, aches, nausea. You may or may not have diarrhea. Chills, fever, and sweating are often associated with it as well. You can have falciparum malaria, where we have the persistent fever, cough. I said couth, but I'm pretty sure it's supposed to say cough, and weakness for weeks, and we won't have any relief. There's also the development of cerebral malaria, where we'll have small blood vessels in the brain becoming obstructed due to the parasites. What I want to say is that these symptoms will, you'll have them and then they'll go away, and then have them again and then they'll go away, and it'll cycle like that because of the organism growing within your red blood cells and how your body would be reacting to that. It's often confused with the symptoms of dengue, because dengue has a very similar symptoms. It doesn't cycle the same, but it can have similar. People can get confused as to what's causing those diseases. All right, malaria caused by parasites in the Plasmodium genus. They are all apicomplexans. So that's what the ones that we were talking about being internal parasites as far as protozoans go. They live in animal hosts. They lack locomotor. Typically, that's where they like their where I'm trying to say, the reservoir would be in animal hosts, and then the actual mosquitoes are what would be transmitting it. We have an asexual phase. That is what the life cycle is within the human. And then the sexual phase is what is going to occur in the actual mosquito itself. The mosquito itself is actually infected by the malaria. Okay. Genera involved include Plasmodium, Malariae, Vivax, Falciparum, Ovale, and Knowlesi. These are all going to be particular to particular areas typically. This is just showing their life cycle. You get bit by this infected mosquito, it gets into your red blood cells, and then it will cycle through of growing in the red blood cells and bursting out, and growing in the red blood cells and bursting out, and infecting and growing in, and so on and so forth. And that can also happen within liver cells. So that's kind of why you have the cycling of the symptoms. Pathogenesis and virulence factors associated with malaria. We have invasion of merozoites into red blood cells. So the merozoites are the parasite, that stage of the parasite that infects the red blood cells. So the merozoites will infect the red blood cells, cause the release of fever-inducing chemicals into the bloodstream as a result. Plasmodium will metabolize glucose at a high rate, so they're going to use up a lot of our cells' food. They cause damage to red blood cells that can lead to the development of anemia as a result. We have accumulation of malarial products in the liver and immune stimulation in the spleen that can lead to enlargement of the liver and the spleen. And then the parasite will undergo a lot of antigenic variation. So our immune system can't keep up with that necessarily. So the vaccine isn't particularly useful unless they can target something that isn't going to change a lot. So there has been some study into development of a vaccine, but so far it hasn't been particularly reliable. And so transmitted by the mosquito, the Anopheles mosquito, pretty common vector for a lot of diseases. It's restricted to a belt around the equator, has to do with the heat and how the mosquitoes like it. And then there are 200 million new cases a year. 90% of those are in Africa. And then there are 500,000 to 1 million deaths annually due to malaria. How do we prevent it? This is really important. And this will be one of the questions that's on the exam. You need to strive for long-term mosquito abatement. We will do that by eliminating standing water in the areas that are affected, spraying insecticides widely, using sterile male mosquitoes that have been genetically altered to be sterile, introducing them into the population so they can't create babies, and then or lay eggs essentially. Then mosquito nets. The mosquito nets themselves can also be like impregnated with insecticide. We also want to, for people in the meantime, treat them prophylactically. So chemoprophylaxis, so treating with drugs. You go to an area with malaria, they will give you prophylactic treatment with drugs that are used to treat malaria so that you don't get it. So people with a history of malaria can never donate blood, just in case there's some still hanging out there and looking for development of a vaccine. How do we treat malaria? We use quinine-based drugs like chloroquine, but now that's becoming ineffective due to resistance. So now we have mefloquine and pyrimethamine plus sulfadoxine. Sorry, these words. Artemisinin, and then counterfeit malarial drugs are being sold in Southeast Asia. These are also contributing to resistance because they're not being taken properly. Ooh. So moving on to HIV. HIV is human immunodeficiency virus. It was isolated in the 1980s. It's a retrovirus, meaning that it takes its RNA genome, turns it into DNA, and puts that into our genome. It causes, over time, can develop into AIDS, acquired immune deficiency syndrome. We have a complex of symptoms associated with infection, including pneumonia, typically caused by pneumocystitis gyrovecii. And then we have Kaposi sarcoma, sudden weight loss, swollen lymph nodes, and general loss of immune function overall. So signs and symptoms continued. We have, the level of virus in the blood is associated with the level of the T cells in the blood, because that's what the virus is going to infect. The initial symptoms include fatigue, diarrhea, weight loss, and neurological changes. The opportunistic infections can set on as this infection is continuing, leading to severe immune dysregulation, hormone imbalances, metabolic disturbances as well. Eventually, we see weight loss, diarrhea, and poor nutrient absorption. A protracted fever, sore throat, and night sweats develop. And we'll see lesions on the brain, on the meninges, spinal column, and the peripheral nerves as infection continues. We can't fight it off. So this is essentially what it would look like during the different stages. I'm going to go ahead and go to the next page. This is a great table to look at while I'm talking about things in the next slide. Okay, so stage 1, we don't have any AIDS defining opportunistic illnesses at all, and T lymphocyte counts are about greater or equal to 500,000 cells per microliter. So not too concerning at that point. Stage 2 of the infection, we still don't have any AIDS defining opportunistic illnesses. We do have a decrease in the T cells. Stage 3 is development of AIDS. We have documentation of opportunistic infections that we do associate with AIDS, and continued decrease of CD4 lymphocyte count, which are CD4 cells, are T cells. These are the specific ones that HIV will target. So I mean, we have a lot of opportunistic infection resulting due to that decrease in the T cells. So that's the development of AIDS. And then other stages, we might have other kind of symptoms or presentation of T cell decreasing. So infection with HIV is caused by retrovirus in the genus lentivirus. So that's the actual genus is lentivirus. It contains a reverse transcriptase that allows it to make a double-stranded DNA from its single-stranded RNA genome. So its actual genome is single-stranded RNA. And then when it gets into cells, it can turn that single-stranded RNA into double-stranded DNA to be incorporated into the host genome. The viral genes will become permanently integrated into the host genome. HIV-1 is the most common form in the world. And then we also have HIV-2, and there are different subtypes of both of those. Important here is that, so HIV itself will interact with a CD4 receptor on a white blood cell, and also interact with a CCR5 co-receptor. So co-receptor means just it's another receptor that it needs to interact with in order to get into cells. So that is how HIV gets in, and that is thanks to these spikes that are on the outside of HIV, and these are the GP120 spikes. So that's over here on the left, if you can see it. Glycoprotein is what GP is. Anyways, essentially the virus is going to get into the cell. Its single-stranded RNA is going to be turned into early double-stranded DNA, essentially. Once we have the complete double-stranded RNA, I'm not making this any clearer for you guys, I'm sorry. The single-stranded RNA genome will be turned into a complete double-stranded DNA bit that will be incorporated into the host genome permanently. Then when it wants, it can make more single-stranded RNA as a just normal production in the cell, and that can lead to packaging and making new viral particles to infect cells. Transmitting HIV. So we have transmission through direct blood exposure through like needle sharing. We have blood exposure during sexual intercourse or intimate contact. And then seminal vaginal fluid exposure during sexual intercourse can also lead to transmission of HIV, even whenever blood does not seem to be present. I'm not going to get too much into this. It's just talking about the frequency of transmission of HIV due to sexual contact, and whether that is due to heterosexual contact or homosexual contact. It is more common to have it transmitted by homosexual contact. That's just the numbers. It's also transmitted by intravenous means. So we have seen cases from blood transfusions, not as much anymore, but we test pretty highly for that. So there's a lot of, HIV associated with drug use, and then you can transmit it from mother to child. You will not get HIV from toilet seats or from mosquitoes. So these are some numbers associated with HIV infection. Well, pretty close, not right now, but pretty close to not too distant past. Right, so a person is diagnosed as having HIV if they have tested positive for exposure to the HIV virus. This diagnosis is not the same thing as AIDS. Viral testing will be based on detection of antibodies to the virus in serum and other body fluids. We'll do that via ELISA, a latex agglutination, which is similar to ELISA, but it's not depending on color change. It's depending on, agglutination, which is like clump forming, and then rapid antibody tests like what we see with what we use for at-home COVID tests. Okay. So yeah, this is just talking a little bit about how the drugs work to treat, I guess, HIV. We have AZT blocking development of the, what you turned into an eraser? Why? Right, so sorry. So we have drugs like AZT, DDL, and 3TC. These are nucleoside analogs. They inhibit reverse transcriptase. This is, I swear. They reverse. Sorry, I'm getting real frustrated now. Anyways, they inhibit reverse transcriptase because they're mimicking nucleoside, like base pairs. That's what I'm trying to say. Base pairs, like in the DNA, so that the reverse transcriptase can't keep going. Okay, the AZT, you can see here, this is the AZT, and this is like saying like, hey, this reverse transcriptase in the yellow can't work anymore because we're on here. So that's how those work. And then we have protease inhibitors that will plug into active sites on HIV protease so that we can't make the proteins that are needed to build the virus up. So that is what the blue block is, the protease that is used to make proper proteins for packaging. It can't work anymore, so we can't make the actual viral particles anymore. And then we also have integrase inhibitors that prohibit that double-stranded DNA that we made from the reverse transcriptase. We made it, but we cannot incorporate it into the host genome. That's what this is blocking. That's what the integrase would do. It would help incorporate into the host genome. So that's how drugs that treat HIV work. This is a little video, really quick, on HIV vaccine development.
There are a number of strategies available to prevent HIV infection. However, ensuring a durable end to the HIV pandemic will require development of a safe and effective vaccine. HIV presents distinct challenges for vaccine development. For example, the virus mutates rapidly and has unique ways of evading the immune system. Vaccine development begins in the laboratory. Basic research on HIV and its interactions with the immune system inform the design of vaccine candidates, which are first evaluated in the lab and in animals. Those that are non-toxic, unlikely to cause serious side effects, and able to induce immune responses that may protect against HIV infection could advance to clinical testing in humans. In early-stage clinical testing, researchers administer the vaccine to a small group of healthy volunteers to assess its safety and ability to elicit the expected immune response. Next, researchers test the vaccine's safety and the types of immune responses in more people. Promising vaccine candidates may advance to late-stage trials that examine safety and the ability to prevent infection in a very large group of volunteers. Historically, many investigational vaccines that initially seemed promising in lab and animal studies disappointed in human trials. In 2009, results from a study in Thailand involving more than 16,000 participants provided the first signal of efficacy for an HIV vaccine. At the end of the U.S. Army-led study, researchers observed a modest 31% reduction in HIV infection among vaccine recipients. Since the results were announced, scientists have been working to modify the design and schedule of that vaccine regimen to try to improve the immune responses elicited by the vaccine. They also adjusted the vaccine to target the HIV subtype common in South Africa, where the prevalence of HIV is among the highest in the world. Nearly 20% of adults in South Africa are living with HIV. Researchers are launching a new study called HVTN-702. This study will determine whether the regimen is safe, tolerable, and effective at preventing HIV infection among South African adults. Approximately 5,400 healthy volunteers will be randomly assigned to receive either the investigational vaccine or a placebo. Neither they nor the study team will know which until the end of the trial. To see if the vaccine works, researchers will compare the rate of HIV infection in people who received the vaccine with those who received the placebo. Final study results are expected in about 5 years. If the vaccine offers substantial protection, making it available to more people would be the next step. After additional studies, scientists may submit the data to regulators for potential licensure of the vaccine. Whatever the outcome, this work will help advance development of an HIV vaccine to prevent new infections and save lives.
So as you can see, I mean, HIV is actually a pretty serious problem in the world, and especially in South Africa. I actually thought the number was higher than that, but apparently 20% of adults are living with HIV. That is an astounding number. So it's definitely something that we need to put a little bit more work into. So, okay, so which of the following is the target of most anti-HIV drug therapy? For most anti-HIV drug therapy, most of them are going to target the reverse transcriptase because it's just nothing like anything that we have in our own cells. So it's a safer target. All right, so to summarize, chapter 20, we talked about how blood and lymph themselves are sterile. The spleen is an important lymphoid organ of the cardiovascular system for filtering out, dead blood cells and things like that. And then we talked about all these diseases that are listed over here on the right with this table, which is a good study tool as well. You might take, something like a picture of a body, and then take all of the diseases, not even just necessarily the ones that we learned here on the cardiovascular side, and pair them with whatever area of the body they might be associated with. That's a good way to look at that. So would help definitely for sure. If you were looking into learning about infectious diseases in healthcare and something like that. So anyways, that's the summary for chapter 20. I'll see you guys in chapter 21. We're getting there.